All right, it's about an hour after the last video where we looked at fertilization. We'll see what the results are. And remember that on this slide, I added a lot more sperm than I normally would. And I ended up getting complete fertilization. And this one has gone all the way to the 4-cell stage. You can count them 1, 2, 3, 4. So has that one right there. This is the same slide we were looking at, by the way. I put a little Vaseline around the cover slip, and that keeps the cover slip from squashing the eggs or embryos, and it also keeps them from drying out. You can see that there's a few sperm that are still hanging around that fertilization membrane even after the egg has divided. So there we are at 4 cells. And this one seems to be a little bit of a late bloomer. This one's just at the 2-cell stage. Guess this one got fertilized maybe a little bit later. But most of these are at 4-cell. And okay, these down here evidently have not divided quite as fast. So these are still at the 2-cell stage. And that fertilization membrane is not going to expand. The embryos are going to stay within that membrane until they develop cilia and the ability to swim, and then they'll break out of it. But they're not going to get physically any larger until, in fact, they have developed a gut and are able to start feeding. I don't know if we're going to be able to feed them in this lab, but I'm going to try to keep some and get them to develop as long as I can. So there we're at the 2-cell. Let me see. That's another view of a 2-cell embryo right there. Yeah, that's a very fine 2-cell with the cleavage furrow right facing you. So we focus up and down on that. And yes, that is a very fine 2-cell embryo. So in the time after fertilization, which has been a little more than an hour, I think by now it's about an hour 20, some of our embryos have, for whatever reason, gone a little bit farther than the others, and they've made it all the way to the 4-cell stage, and some have only divided once. But so far, they all look pretty normal. Although this one is actually not, because that looks like a 5-cell embryo with a distorted fertilization membrane. That ain't right. Remember, when you add too many sperm, you can end up with an egg getting fertilized multiple times, because the more sperm you add, the greater the odds that 2 of them will just happen to penetrate at exactly the same moment before the blocks can start. And when you end up with a double fertilized egg, or sometimes more, you can end up with weird cell division patterns because, of course, the cell is not diploid. The chromosomes can't divide evenly. And yeah, you can get pretty non-standard, downright weird patterns at this stage. And let me see. So that one is not doing so well. Let me see if I can find any more. Yeah, this one might be 8-cell, but there's something odd about those cells. They're not all the same size, and that's not right. They should be all the same size. at these early stages. So this one may not be dividing evenly because it's got a whack number of chromosomes that it can't figure out how to divide through mitosis. So that, kids, is why we always dilute the sperm. That one may have, this one up here, 1, 2, 3, 4. I count 6. And again, that's kind of odd. And this is another one that I think got fertilized too many times and is having to cope with exactly the wrong number of chromosomes to divide properly. So yeah, some of these are, yeah, this one right here is another looks like a 5-cell. Yeah, I can see 4 on top and then there's 1 underneath. So again, that's what you get if you bomb the eggs with too much sperm. You get abnormal development. Okay, some of them aren't so bad. That's a very pretty 4-cell embryo, but we've seen some weird ones that you can get from double fertilization, and that's why those mechanisms are important. Okay, here's what I've done. I took the rest of the eggs and I fertilized them with pretty dilute sperm, I hope, in a couple of large dishes. Each one holds about 1.5 liters, and I'm going to leave them overnight. And one's a control, and one of them has had some nickel added, and nickel is known to be embryotoxic. We added a very small concentration of nickel, but I'm going to check these out tomorrow morning when hopefully you're all here, and I'm going to check them again tomorrow afternoon, and we'll see if we can detect any effect. Hey, that rhymed. We'll see if we can detect any effect. Okay, I lost it. I was actually starting to get some flow, and then I blew it. Never mind. I'm tired. We'll see if we can detect any effect of nickel on early development of sea urchin embryos, and that's going to be the experiment that we do. So with this beautiful 4-cell sea urchin embryo that we produced here in the lab, I'm going to sign off for the evening, and I will catch you folks tomorrow at 9 o'clock, same time, same bat channel.